Hello, and welcome to today's webinar, Life After COVID, Positioning Your Lab for Success in a Post-Pandemic World, hosted by Dark Daily and sponsored by Ovation. My name is Liz Carey, and I'm the managing editor at the Dark Intelligence Group, here with Amanda Curtis, who heads our webinar technical team. Before I introduce today's presenters, I want to mention that we look forward to answering your questions. Just use the questions box of your GoToWebinar control panel. And again, we want to thank Ovation for sponsoring this event. It is now my pleasure to introduce our speakers for today. Dr. Barry Wark is CEO of Ovation, a clinical informatics company focused on redefining the role of molecular laboratories in delivering better health care. Next is Dr. Jamie Platt, CEO and founder of BRIDGenomics, and Jamie has more than 15 years of leadership in next-generation sequencing and clinical diagnostics. Also with us is Dr. Andrew Warren with Third Rock Ventures, and Andrew has a biomedical engineering background, and you could read more about Andrew here on the slide. Together, this panel not only brings together decades of experience in genomic testing and lab operations, but also practical advice for how clinical labs can begin to prepare themselves for a post-pandemic world, and that's something that's on all our minds. So to get us started is Barry Wark of Ovation. Barry, tell us what's on your mind here with COVID-19 and the surge in molecular testing volume.
Sure. Well, it's a pleasure to be here, and it's an honor to be here with Dr. Platt and Dr. Warren. So thank you for both of you for joining us and for everyone here today. What's on our mind is that the coronavirus pandemic created a need for a dramatic increase in infectious disease testing capacity throughout 2020. In the US alone, more than 343 million COVID tests have been performed. And the vast majority of these tests have been performed in private laboratories, with the rapid rise in testing spread broadly, at least proportionally, between small specialty labs all the way to large reference labs. So these labs, along with our entire medical infrastructure, had to ramp up capacity dramatically and quickly, and now face some challenging business decisions about how to continue to grow their business as vaccination numbers increase and as the pandemic hopefully subsides. So what was the experience of this like for diagnostic labs? Dr. Platt, I'll let you take it away.
Great, thank you, Barry. Well, labs certainly have faced all sorts of challenges this past year, and as you mentioned, there's a lot of different types of laboratories. So we may be talking about existing molecular labs that were in existence pre-COVID. We may be talking about labs that were not doing molecular testing pre-COVID, but now are. And then there's also the startup labs. So a number of labs formed specifically to address COVID testing needs. And that may include mobile labs and pop-up labs. So I wanted to provide that bit of background because some of the data I'll discuss has to do around several categories of laboratories, of which those I just mentioned fit into commercial reference labs, academic medical centers, community hospital or health system labs, as well as public health labs. So on the next slide, let's consider the impact that this race towards molecular testing for SARS-CoV-2 or COVID has had on clinical labs. So there's been a race to adopt and expand molecular testing capabilities, as you're all well aware. And let's start with this simple statistic that for at least 48% of clinical labs have adopted new testing methodologies or automation in order to meet the demands of COVID volumes. So first, this created, as you are all painfully aware, this created a huge supply chain issue. So in April, the Association for Molecular Pathology, or AMP, survey reported that 85% of laboratories experienced a significant delay or interruption in testing due to the inability to get supplies. The August survey on SARS-CoV-2 diagnostic testing reveals that 90% of labs have experienced delays and or increased testing due to issues. So this means that the supply chain issues have not resolved. While we've seen a shift and maybe better supply chain or supply availability on swabs and viral transport media, there's still a significant issue around molecular test kits, reagents, consumables, and even automation such as liquid handlers. So what these supply chain issues have done is really forced many labs to expand their testing methodologies, especially as kit and instrument manufacturers imposed restrictions on allocation of testing kits and reagents. 68% of all labs have experienced restrictions or allocation issues with testing kits and reagents. So that forces them to shift to another methodology, another test kit, or even other consumables. What this means is that labs have had to learn how to either do a move outside of a standard EUA verification and actually perform more of a laboratory developed test validation, or they've had to bring in another EUA method that can help them have that capacity. So those with R&D organizations or more technically savvy folks on the team can actually, pivot a little bit better because they have the wherewithal to bring up other methodologies or even develop an LDT. However, others still struggle and tend to bring in lower volume tests, maybe that have a rapid turnaround time, but the throughput is not as good. So it's interesting that the April AMP survey revealed that labs had deployed multiple testing methodologies. So commercial reference labs were primarily using one method, whereas academic medical centers and community hospital labs were predominantly running 3, 4, or even more methods. So the number of labs now using only one method in the August survey has declined from 29% in April to now 11%. So labs have been forced to bring in a lot of different methodologies to meet the demand. So this means that labs spend more time training, they have to manage multiple workflows, they have to manage a lot of additional inventory, there's a lot more documentation required, and of course there's more susceptibility to increased analytical errors. So this trend towards a faster turnaround time tends to be lower throughput, but it still has some of the typical burdens of managing the workflow. So let's actually look at the next slide, which helps us understand the shift to multiple COVID molecular testing methodologies and what that means for overall volumes and volume mix. So we can see that while routine testing has dropped significantly for most labs to 77% of pre-COVID volumes, the vast increase of COVID molecular testing equates to about 40% of all volume and brings the number up to 245% above baseline. So labs are really stretched thin with trying to accommodate this volume. Some of their existing menu obviously has much lower volumes. They've had to shift staff, and then keep in mind the volume mix from the workflow perspective may not actually be that different and has increased because labs are having to bring in additional methodologies. So the workload for the lab has been much greater with this increased testing demand, and there's also a lot of other elements the laboratories have to manage around information flow. So aside from inventory, the quality systems, documentation, there's also, as everyone is aware, a reporting requirement to local, state, and federal governments, which has really pushed labs to understand that they are in the information business. They have to be able to utilize their own information, flow their information appropriately throughout their organization. So let's go ahead and move to the next slide. And we can see that what this does is really has a major effect on lab staffing. So it's actually very difficult to get enough staff to run some of these tests. So folks have tried to move to automation, But obviously with supply chain issues, even if folks could get a automated liquid handler, which was difficult to get and still is in many cases, there's an issue with the pipette tips or other consumables needed for those systems. So lab staff has been a huge issue, not only in terms of needing more folks, especially when we can't implement automation, but even when automation can be implemented, finding staff that can manage the automation and can handle the automated systems can be very difficult in some settings. I think the other thing to consider is that, again, about the information flow, and when we think about now we have to have staff that are trained not only to pipette things a certain way, but they have to manage being able to think about working with their limbs, maybe working with interfaces to patient testing scheduling, and then, as I mentioned before, how those results not only get reported out to the physician or to the patient or to the agency, but how do they get reported out to the various entities involved in HHS reporting. So let's go ahead and move to the next slide. What this means is that folks have really ramped up. Laboratories have done an outstanding job in meeting the demand, and they've really rallied around the need to increase their capacity. They've had to do it with multi-pronged approaches, but there's still a lot of uncertainty about the future. So now that they've implemented these, sometimes these systems, maybe some automated systems, now that they've brought in maybe a new molecular device, or in some cases, several new molecular instruments, what are they going to do post-COVID? And when will that post-COVID time really hit? I think a lot of people were expecting we would see a downtrend in COVID testing by this point, but in fact, we're not. And even the director of the CDC is very concerned that people are not, social distancing and wearing masks. And we've seen an uptick in the recent 2 weeks in COVID cases and COVID hospitalizations. And then along with the presidential administration's move to reopen schools and get the economy back on track, it's still not clear what the future of COVID testing will look like. So let's go ahead and move to the next slide. And what I'd like to do is just ask Andrew to speak a little bit about how this also then impacts life sciences. So I've spoken a bit about supply chain and things that affect the laboratories, but obviously this is a network of different industries that have to work seamlessly together in order to really meet the needs. So thank you, Andrew.
Thanks, Jamie, for the lead-in. I think there are a lot of really interesting opportunities and overlaps, particularly how the world has changed in the last year or so, both as relates to COVID, but as relates to better understanding COVID biologically, as well as how this might change the overall perception on what drug development looks like and what role labs and biomarkers can play in this. Next slide, please. So I'll talk a little bit about short-term impacts and then talk a little bit about focus on other pieces of COVID that maybe we haven't thought as deeply about, or what might be coming through from a drug development perspective. So the main bullet here is, what near-term catch-up do we need to do in the biopharmaceutical industry to account for the broad disruptions from COVID? This catch-up is to figure out how to fix studies that have been interrupted, how to bridge them with data that maybe doesn't have all the time points or data points that we want, particularly for the development of drugs that don't treat acute life-threatening diseases, which were much more likely to be interrupted due to COVID. There's an interesting type of short-term regulatory flexibility that goes along with this. So next, we'll talk about how more pragmatic people have been. People have been hoping for remote care and remote recruitment, measurement of biomarkers, separation between integrated clinical trial sites. And it took a pandemic to catalyze that transformation. But for many parts of the industry, I think people won't be comfortable going back or won't be interested in going back. So there's a huge emphasis on being able to move more quickly with these studies. And some parts of that haste, I think, will be maintained as we go forward, as well as different ways of collecting samples and driving testing. With vaccines, it's clear that mRNA is the future for development, at least of this type of vaccine, particularly as the variants develop. But there are also other opportunities for other type of mRNA drugs to impact development. And with that comes a more quantitative, programmable type therapeutic. That's something the field has been pushing for some time, but we're starting to see a huge change. For labs in particular, emergency response will continue to be incredibly important, as will vaccine response speed. Maintaining emergency capacity to understand how to deal with variants as they crop up, as well as potential future pandemics, is going to be on people's minds. And while we do all tend to have short-term memories, I think it's important that maintenance of this emergency capacity will be important, both for, again, variants of COVID or other future outbreaks. Another interesting thing has been that pragmatism beyond recruitment and trial management has also extended into real-world evidence. Some of this has been very positive, some of it has been a lot of noise, and it's difficult to tell them apart, but people are getting a lot of practice at rapid iteration here. I think there's a role for that type of real-world evidence data to be meaningful at scale and to allow us to answer questions that otherwise would have to be done with huge, long, slow trials. And then finally, on infectious disease, it's to be determined how long the attention span will remain, but we can hope that this will increase people's interest in monitoring, tracking, and understanding infectious disease, as well as development of therapeutics. Along with that, there was increased interest in understanding it, right, in developing assays or monitoring the impact of these diseases, which I think will hopefully have knock-on effects throughout the industry. On the next slide, I can talk a little bit about long-haul cohort, which is more directly COVID related, but is going to be important for the field to be mindful of. So an estimate is 10 to 25 million Americans will be affected as long-haul COVID patients. Because of that tremendous need that so far there hasn't been a concerted effort to understand and monitor that. It's going to be really important that the diagnostics are able to track and understand that, as well as access to these patients and their existing samples. There are going to be a lot of interesting cross sections of long-haul COVID patients, and the biology still is not fully determined. So to maintain interest and to facilitate rapid development to these people who are in great need, there's more thorough characterization as well as monitoring of how these people respond. For example, there's emerging data that vaccination may be meaningful to a subset of these long-haul COVID patients. How can we use existing real-world data or samples that are available to help validate some of these findings? It's not a problem that's going to go away overnight, and because of the difficulty and intractability of the problem, there are actually a lot of similar diseases that for a long time have been underneath the radar, simply because they're so hard to quantify. On the next slide, I can talk a little bit about what research can do to improve our ability to understand these patients. So a substantial portion, 62% of severe COVID disease, presents as chronic comorbidities. So many of these patients are not healthy, and there's a wide variety of comorbidities that can worsen COVID outcomes. Some of this may also interact with the long-haul cohort, but it's going to be crucially important that labs and data sets are able to account for this, both to deconvolve what role was COVID and what was the underlying comorbidities, to look at the dynamics between these diseases as they develop, as well as accessing very specific subpopulations of these folks to understand that specific part of the disease in detail. The improved appetite for real-world evidence, as well as huge existing availability of banked cohorts, will be very meaningful going forward in the future. And a lot of it's up to the industry to be thoughtful on how to facilitate this research. So on the next slide, Jamie and I will talk a little bit about what are the interactions between these changing developments in clinical labs and drug developers, and what are the ways that the field can collaborate in order to move forward. So again, I mentioned these huge new patient cohorts. It's going to drive drug makers and innovators to be interested in accelerating development. How can we get our hands around these subpopulations? Because there's a lot of diversity going on in the way the disease presents. And honestly, we need to look forward with humility on how much we understand the biology of COVID, as well as the emerging variant data. There's going to be a huge availability of real-world evidence where people are incentivized to take advantage of this. There's an appetite to maybe move a little bit more quickly than there otherwise would be. And then finally, my hope is that trials are going to be run differently going forward. Some of it will be subtle at first, but the distribution and how samples come in and how monitoring on patients changes is going to be something that hopefully is lasting and will increase the rate of development. But it's up to both fields to collaborate to move that forward. So I'll let Jamie maybe circle back to some of the parts on clinical lab where these two fields can work together.
Great, thank you, Andrew. Yes, I think, the first thing that comes to mind and is highlighted here is really the value of the samples and creating a valuable sample flow. The data and information that we can get out of these samples are extremely valuable. And again, this brings us back to labs thinking of themselves as information providers. So what does that sample sitting in a freezer or a sample that's about to get discarded really mean in terms of the value of data and information? So that's something important to consider. And I think we've also seen with the move to quickly adopt new technologies, put multiple new molecular technologies in place, labs are going to want to be able to utilize the resources they've put in place and monetize this in a post-pandemic world. And some of that will be around how they've developed better R&D capabilities, how they've strengthened their quality systems as they've had to validate or verify more tests quickly, and then again, how they've had to train and bring on staff that can deal with information and are more informatics minded. So labs with R&D capabilities and with informatics capabilities are poised much more beneficially in this space. I think they have an advantage to be able to leverage the information and the skills they have to work with drug developers to do maybe these biomolecular and biomarker studies, as well as looking at vaccine efficacy studies and other elements that are going to be needed. And we've already seen that around next generation sequencing and variant detection. So with that, we can talk a little bit more about, we can go to the next slide and talk a little bit more about what this means for labs. So what can labs do now, and what are they doing now that is creating a valuable asset for the future? So what we see is that these I guess before I go on, I think what I'd like to do is suggest that we stop for a poll question.
Yeah, Amanda, let's take a look at a polling question because we have so many people on the call today, which is fantastic. And these questions may inspire more specific questions for you for the potential, for the panelists about potential barriers. But does your lab have a plan for utilizing its leftover COVID samples? And all you need to do is select yes or no. And we'll keep an eye on the survey responses, and we just have a few seconds. Let's see. All right, going to take in a few more before Dr. Platt continues. right. Okay. Dr. Platt, do you want to comment on where we're headed with the poll, and we're about ready to close the poll, everyone?
Great.
Let's go ahead and close the poll now.
Excellent. So do we have, so I guess, yeah, sorry, go ahead.
Yeah, absolutely. It looks like a majority of those who have answered the poll do not have a plan for utilizing their leftover COVID samples. So what are your thoughts?
Yeah, that's quite interesting. So I would imagine that has to do with several factors, and only those on this webinar that answered the poll would be able to specify why that is. But as we think about the mix of laboratories that are collecting these specimens, there is a huge issue around differences in informatics capabilities and how that data can be stored or moved around the lab. There's also infrastructure considerations. So, you know, for labs that are doing a lot of high volume in COVID sampling, it's difficult to maybe store many of those specimens. And so, especially as we've had supply chain issues with regard to freezer and specifically freezer space, that can be very challenging. I think the other thing to consider is labs are also doing a variety of different types of testing on different specimens. So for labs that have moved to maybe extraction-free saliva, or to maybe direct swab methods from anterior nares or others, storing those samples in a reliable way could be challenging. So I'm sure those who answered the poll have a variety of different reasons that maybe they haven't thought about it, or maybe they've thought about it and assumed it's too difficult. But I think what I'd like to point out is that there's a wealth of information in each of those samples, and as we look to, you know, help other, you know, bring on new tests, even within our own labs, having access to those specimens and those samples is critically important. It helps us develop the new technologies, the more streamlined technologies that help with throughput and capacity, helps us understand variant differences, and then, as Andrew mentioned, really helps us be able to tie and leverage that information into other business opportunities and monetize the information that we're sitting on when we have a sample in the lab. So I would challenge folks to think about that specimen in the lab as being information, and information that's valuable to vaccine developers, therapy developers, as well as to, you know, folks doing surveillance and folks looking to validate other methodologies that, you know, help us get to answers more quickly. So I think we can move to the next slide. And pretty much, you know, I've really already discussed, you know, how we should think about being able to leverage these as strategic assets. So let's go to the next slide and really think about some specifics here. So when we think about the value of information in our specimens, I think it's important that we all consider how we may need to update the consenting strategy. So many labs are using remnant samples, and that's fine in a CLIA setting. But as we think about how to use the information and these samples in a broader sense, it's important to think about what type of consenting we need to have for follow-on research. So let's go to the next slide and talk about how, again, we use these remnant samples that can be de-identified, how we go about de-identifying the samples to begin with for that use, and then how do we want to be able to, you know, have folks, the subjects that have provided these samples, how do we want to be able to access them perhaps for other research activities? So there's a lot, again, about the information and how we flow the information, how we keep the information private and secure, and how do we really make use of this of these specimens that we're sitting on. So it's important to think ahead about how we do that consenting, the type of information we collect on a requisition, for instance, as well, because the more clinical data we can collect at the time of the requisition, the more valuable that information is. Let's go ahead and go to the next slide. So the other thing that's important to think about is really to identify those high value samples. So what samples have the best material, thinking about not only variants of the SARS-CoV-2 virus, but also maybe specimen type and stability. What types of samples really hold the most value, not only for the testing we're doing in the lab or for further validation, but for biomarker studies or working with folks outside of the lab industry, the life science companies, the biopharma, to really help think about how we can get the most value out of the work that's already been done. So let's go to the next slide and think about how this plays in when we think about all of the testing done in the laboratory. So I think it's been pretty well shown in oncology testing how important having certain types of variants and samples can be when additional validation or new clinical information and insights come up. So whether folks are sitting on SARS-CoV-2 samples or whether they're sitting on more of their routine testing samples, thinking about the sample value and what that means to drug development pipelines, vaccine development pipelines, and other biopharma activities is really important. And I should also mention that it's also important to device manufacturers and instrument manufacturers that are trying to think about how to uncover certain types of variants or detect certain types of biomarkers in different types of specimens. So sample quality is, of course, an issue, and then storing that sample in in a way that retains that sample quality and stability is really important. But most labs nowadays are really flooded with a lot of these different types of SARS-CoV-2 samples, and it would be really good if we could make use of that and work together as an industry and across industries to help bring the value from that. So let's go to the next slide. And the other thing, that is on a lot of laboratory managers' minds is, what are we going to do now post-COVID? Well, I think it's been surprising to many. I think most thought that the COVID testing volume would be trending down significantly more than it has been to date. So we are going to continue to see incoming samples for COVID testing. And I think it's not too late to think about a longer-term strategy on how you deal with the samples and, more importantly, the information. So for those labs that have managed to move really quickly to put good informatics in place that help with reporting to the local, state, and federal agencies, for those that have started to think about how they manage their information, those labs are in a really good place to really move beyond thinking about the sample and thinking about the information, how to begin to leverage the information they have from the samples in the broader life sciences space. So in the next slide, let's talk about a few issues before we take another poll. And those would be, labs are now faced with some really critical decisions. Do they stay in the COVID testing space? And certainly some of the larger reference labs or labs that were previously maybe not even doing any infectious disease testing, they'll have the decision point of whether or not they should actually even continue to do COVID testing, or whether they offload that to other labs and decide to focus on their core business strategy, either around oncology testing, genetic testing, or whatever other models they have going on. So I think larger labs that brought COVID into just help alleviate and do a public service are going to have a very different decision than those labs that have either started new specifically for COVID, or maybe even the labs that brought molecular testing on to help meet the need, but haven't traditionally been a molecular testing lab. So some big questions for labs, and it's not an easy question to answer, but I think if we start to look at the value of the samples, the value of the information, and then even beyond COVID, thinking about when we all get back to our routine testing, whatever that menu looks like, how do we continue to leverage the samples that come into our lab day to day? I think COVID has really taught us, when we look at the telemedicine reports and others, that people are really more comfortable with information, and there's a push and an opportunity now to really, for labs really to look at themselves as information providers and to build business strategies and solutions around more seamless integration of information. So I sort of already touched on the fact that they would have to reevaluate their testing menu. The large labs that weren't in the infectious disease testing space will probably take a different direction than those that have had a broad menu. And then again, considering how to move that information model, pair it up with all the activities in telehealth, and then also in direct to consumer. I want to just mention really quickly before we go to the poll that there was a really interesting paper published on March 28th in the Wall Street Journal by Laura Landro, and it was highlighting what the telemedicine in the COVID era and what we've learned from that. So what we've learned, what works, what doesn't work. And the interesting statistic from that is that it really seems that 20 to 25% of all healthcare in the future could be delivered remotely. And that's a major push from what it was pre-COVID. So in one of the surveys in that report, close to 75% of all patients who completed a telemedicine visit would actually prefer to do a telemedicine visit again over an in-person visit, even post-COVID. So with that, I think we are going to turn to another poll question.
Yeah, Amanda, let's launch that next poll and hear from the audience on this one. If your lab's COVID samples could be repurposed for life-saving research, would you take the additional steps to preserve them? Simple yes or no there, and we'll give about a minute for everyone to complete this poll question. And we'll give it another moment or so. And I want to leave enough time for questions and answers here at the end of the program. So, Amanda, why don't we go ahead and close this poll now? And we should be able to see the results. As you can see there, it looks like repurposing the samples is a possibility. And so there may be some considerations here that our next speaker could touch on or answer some questions about.
Wonderful. All right, so let's move on to the next slide. So I think from that poll we can see that, previous slide. I think we can see from that poll that there's a lot of interest in this moment and in the ability for labs and life sciences to work together. So Jamie and Andrew, thank you for sharing your insights on both the drivers and the impacts from COVID-19. So in this section, I'm going to describe 3 challenges that labs face in making the most of this rapid rise in testing volume from the last year, combined with this new collaborative environment between labs and life science, how they take advantage of that to grow their business and help patients. We know firsthand about these challenges because Ovation has helped labs overcome them. And today I'm going to try and share all of those secrets so you can do it as well. Next slide. Number one, as Andrew described, life science researchers are going to need to do extensive research in order to understand the biomarkers associated with COVID-19 infection and downstream sequelae and morbidities. The leftover samples sitting in labs today are valuable resources for that research, and those labs that hold them could provide valuable answers by doing additional testing of those samples. The first challenge that labs face is that they may not have the right specimens available for a particular research project. So a researcher may be looking to measure a specific biomarker in a defined cohort of patients. And if your lab doesn't have the samples that can be measured for that biomarker, or doesn't have enough samples to fill the full cohort, then your samples aren't valuable for that researcher for that project. This sounds like a contradiction. I just said those samples are valuable resources, and yet they may not be valuable for a researcher. And the resolution of that is that the samples are valuable because there's a lot of research to be done, but matching the right samples to the right research project is a challenge. And it's a challenge for labs looking to commercialize their assets because they need to find projects that match their sample pool. So labs can tackle this in two ways. First, they can enrich their sample flow for a particular cohort or therapeutic area. This is some of the actions that Jamie was describing. Labs can adopt new technologies or new assays and the laboratory workflows that go with them in order to go to market and build a book of business for that therapeutic area. And that may involve, acquiring new customers, new providers. Second, labs can work together as a collaborative network to combine their leftover samples for projects, right? In this collaborative effort, the aggregated set of samples is able to service a much broader range of research projects. Next slide. Number two, the clinical information received by a lab at test order, along with the leftover specimen, are unfortunately often not sufficient to fully answer a research question. For example, it may be hard to determine if a sample meets the inclusion and exclusion criteria for a study based on the lab's proprietary information alone. Linking that specimen to the rest of a patient's clinical journey, of course, increases the value of that specimen. In fact, the value of a linked record that contains laboratory biomarker measurements and diagnostic results linked to the clinical journey of a patient, that linked record is worth far more than the sum of its clinical and biomarker measurement components to a researcher because it allows them to answer a complete question. The challenge is, how do you do that? Well, this linking can be performed in a privacy-preserving manner that uses only de-identified data through a technique called tokenization. So tokenization is an algorithm that generates a cryptographically secure and unique code, or token, to represent each patient. Multiple data sets that are tokenized with the same algorithm can be matched using this unique token without re-identifying the patient. This is how data from your lab can be matched and linked to external data sets in a privacy-preserving manner. The challenge is that properly de-identifying and tokenizing data and samples in your lab requires some significant technological investment. Jamie was describing the work that many labs have had to do to improve the informatics capabilities of their lab for public health reporting and management of larger volumes of data. This is another lift to add the capability to do de-identification and tokenization. And it may further require an expert determination of both your lab's process and of any of those combined data sets in order to adhere with HIPAA requirements. Next slide. Number three, to generate new business from your carefully saved and carefully tokenized specimens, a lab needs access to researchers that want to use that data. We found that very few independent labs have anyone dedicated to life science business development. And this is a role required to both get that new business and to coordinate the research projects associated with that business. One way labs can address this challenge, again, is working as a collaborative network to share some of those go-to-market and business development and research coordination resources. So Ovation can help your lab address these three challenges to growing your business, and these same challenges in helping patients through research collaborations. In fact, Ovation was founded on a vision to make this kind of laboratory life science collaboration possible. Next slide. Ovation enables labs to achieve ambitious healthcare objectives and to grow their business. We produce a LIMS system purpose-built for clinical and molecular diagnostic labs. We have out-of-the-box workflows for cancer genomics, pharmacogenomics, infectious disease, non-invasive prenatal testing, and others, and support for cutting-edge technologies and workflows, including next-generation sequencing, digital and quantitative PCR, and microarrays. Next slide. Molecular diagnostics isn't just the wet lab workflow, though. So Ovation supports the full cycle of lab functions to take a sample from order to diagnostic report, to delivery of that report, reimbursement, and all of the operations that support that cycle within the lab, including operational intelligence and quality management. Next slide. On top of that, Ovation is a full informatics ecosystem for your diagnostic lab. So we have turnkey integrations with electronic medical records, customer relationship management systems, revenue cycle management, clinical interpretation, and a range of instrumentation. With this full stack environment, Ovation can help labs scale up their business, for example, for infectious disease testing, or add new lines of molecular testing effectively and efficiently. This is part of the recommendation that Jamie was presenting, right? Labs may need to go build new lines of business, new assays, in order to take full advantage of this environment. But I think what truly sets Ovation apart is that we can help you grow your lab's top-line business by partnering with our Ovation Research Network. So next slide. The Ovation Research Network is a turnkey program to help labs grow revenue and eliminate downtime by leveraging leftover sample volume and testing capacity. Exactly the kind of things that we've been talking about today. So next slide. How does it work? Ovation Research Network participating labs adopt a universal patient consent that includes the entitlements that Jamie described. And these labs contribute leftover specimens and de-identified data to the network. Ovation handles sample and data de-identification in accordance with HIPAA. We handle specimen transport logistics and biobanking, some of those infrastructural challenges that others identified. If your lab already has appropriate biobanking facilities on site, Ovation can contract with you to store your leftover specimens in your facility. Life science customers ask Ovation's Living Lab to answer questions such as, what is the value of a particular biomarker for patients with a particular disease? To make those biomarker measurements, Ovation finds samples that match the desired cohort and sends those de-identified specimens back to participating labs and pays them to do additional research testing on those specimens. Ovation then aggregates the data and delivers an answer to the customer. In essence, the Ovation Research Network and participating labs become a global distributed biobank able to service non-interventional clinical trials across a broad range of therapeutic areas to benefit patients, the labs, and the life science industry broadly. Next slide. So, the value to your lab for participating in this research network. Well, we've talked about some of the challenges labs face in being able to take advantage of this moment. By participating in the Ovation Research Network, you get access to new markets, right? By aggregating data and specimens together, network participants are able to service non-interventional studies across a broad range of therapeutic areas, right, to address this entire market altogether. Second, labs participating in the research network generate additional revenue. They get paid to do research testing on these leftover specimens. And finally, for labs that are doing internal R&D, network participating labs get access to the aggregated and de-identified data of the network for internal assay development and validation. Next slide. What is most exciting about this research network, and the reason we built Ovation, is that it's a chance to do well and to do good. Participating labs generate additional revenue. That's the doing well part. And together, we're allowing life science researchers to complete non-interventional style trials 10 times faster than traditional trials that create a primary repository. That's a truly massive acceleration, and it increases the ability of those researchers to bring life-improving and life-saving therapies to market faster and more efficiently. That's good business, and it's good for the world. So next slide. Jamie, if a lab wants to take advantage of this moment and what comes next after COVID, what's their take-home action plan from today?
What I would highlight is, consider updating your consenting strategy. That's really important for this and for using these valuable specimens. I would definitely think about identifying your high value samples and consider carefully what you choose to discard and keep. Of course, everyone will be reevaluating their testing menu as we move into post-vaccine COVID era. And then, again, as is typical, labs have to continue to analyze reimbursement. So this really, all of this goes together to feed into how a lab would consider a new model.
And call Ovation, we can help.
Looks like we have a few questions from the audience if the panel is ready. Everybody there, yes. So tagging back to Jamie, you just mentioned, it sounded like consent. So what kind of consent is needed to save these clinical samples and sell them for research purposes? That's a question from the audience. What are your thoughts?
Great. That does vary by the study and the clinical study. And the current situation is that CLIA labs can use their remnant specimens for validation of analytical studies, but to actually use these specimens for clinical research, special consenting is needed. And so that's a very nuanced and complex question in terms of the details that go into that. But that's something that folks should definitely be exploring and thinking about what types of consent language would need to be put in place to leverage these specimens for actual clinical studies.
All right, thanks. Here's a good question. Are the positive samples more valuable than the negative samples? Andrew, is that something you could help us with?
Yeah, I can give a preliminary answer. I think the answer is maybe, but it really depends on the scenario. And so it's important to be open-minded to what the different research questions that could be asked are. Not all research questions are related to COVID, and so that's an important thing to remember. And even if they are focused on COVID, you can imagine there are a few different interesting populations that are more valuable by being negative. The important thing to remember is we don't know all this in advance. And so having a broad perspective on the types of questions people are answering is important. So you can imagine particular value in a negative sample on being a match for a positive sample to make sure you have a really well-controlled study. That's a huge benefit of just size. If you're looking at non-genomic data, there might be an important time component. If you want to look at evolution of a response, even between a positive, negative, positive type sample result. And then you can imagine looking for protected populations as well. So it's important to be thoughtful about what scientific question you'd be thinking of.
Very good. Now, I've had a couple of questions that tag back to that. idea, that market for serving the COVID-19 long haulers, excuse me. So Barry, what are you learning about this market for serving the long haulers and the value that labs could have in this, to work in this space? And then I received another question somewhat similar about certain kinds of institutions that could possibly be the ones dominating this. So how can the labs all get a chance to be at the table here?
Yeah, those are two great questions. So it's hard to say right now what the diagnostic market is for long haul COVID patients. There are 3 to 4 sort of emerging biological explanations to explain long haul COVID, and we would presume that whichever one turns out to be correct includes some monitoring of those patients to direct therapeutic treatment. 25 million patients getting even a couple of tests is a relatively large market, and we would expect that some labs are going to get particularly good at doing diagnostic testing for long haul COVID and presenting that information in a useful way to providers. That's not the only value that we've discussed today, though. The other side of it is the research side. And of course, distinguishing amongst those multiple biological explanations is going to require a lot of research testing. And having primary samples from patients throughout the journey from pre-infection to post-infection long haul sequelae is going to be really important to that effort. You can't go back in time. The sample is gone. And we know that, this prior to COVID, the US non-interventional biomarker study market was multiple billions of dollars a year. More than 70% of each of those individual trials goes to patient recruitment and testing costs. And that's something that labs that are holding samples are able to address, right? Like, it's a lot of dollars for collecting the samples and doing the research testing. And we presume that there's going to be a significant increase in the amount of that kind of biomarker testing going on, biomarker studies going on, to try and understand, as Andrew said, the relationship between long haul COVID, the other comorbidities, and the several biomarkers that are shared between those two populations.
Mm-hmm. Well, sort of along those lines, has there been any instance where these samples shouldn't be used for genetic study, should not be used for genetic study?
Yeah, I can tackle that one. I mean, the really clear one is what we've already discussed, patient consent, right? Once de-identified, these samples are somewhat out of HIPAA jurisdiction, but the absolute right thing to do here is to be very upfront with patients in explaining the secondary research uses of those samples and the benefit that can be gained from them. And what we found is that with a very simple statement that in addition to the testing that's being performed from a diagnostic perspective, their de-identified samples may be able to contribute to understanding this disease and helping people who suffer from it. A lot of patients are quite willing to allow those samples to be used, especially if they can be de-identified and unlinked from the patients.
I think that you just answered another question that we have had on, can the consent be part of current disclaimers? Or should be different forms or be different forms provided? It sounds like people these attendees need to review the language in their consent forms. All right. We had another question, which was, is the Ovation limbs, do you have to use the Ovation, excuse me, we have so many questions. I'm scrolling through and trying to strategize at the same time. Do we have to use Ovation limbs to be able to participate in the research network, in your research network?
No, but it makes it a lot easier. So the Ovation limbs includes a number of the sort of informatics capabilities that Jamie was describing, helping labs organize their samples, prepare them for shipment to our biobank, de-identify them correctly. So there's a lot of capabilities that are built into our LIMS system, make it much easier, but it's not a hard requirement. Give us a call if you'd like to participate anyways. The research testing that happens in the Ovation Research Network does all go through Ovation LIMS, but we can provide that for labs doing that research testing. That's how we manage aggregation and normalization and all sorts of informatics challenges associated with distributed research testing. Got it.
So we didn't get to a lot of the questions, but these panelists will be prepared to help offline with that. And so, we've had some great presentations. The questions, the discussion's been awesome. We want to thank Ovation for sponsoring this event and our panelists from Dr. Platt, Dr. Warren, presenting, and Barry Wark as well. Please be on the lookout for an email with an important survey and on-demand link for this program. So for that, we will, at this time, we'll sign off from Dark Daily, and we hope everyone has a great afternoon. Thank you.